The physical basis of inheritance of alleles is really based on how chromosomes are passed on. We looked at meiosis and mitosis, and we talked about inheritance patterns, normal Mendelian inheritance patterns, variations from that, linkage, polygenic interactions. But everything we've been talking about up until now has had to do with sort of normal chromosomal segregation to the gametes. But what happens when that goes wrong? And the process can go wrong. So our next topic is, we're going to talk more about chromosomes and some of the terminology that goes along with chromosomes. And what happens when chromosomes go bad? So chromosomal abnormalities. So variations from normal chromosomal inheritance or normal chromosomal shape. So let's introduce some terminology and conventions. So when we're speaking about the chromosomes, remember, in humans, there are 23 unique chromosomes. Different species have different numbers in a haploid set of chromosomes, but in humans, it's 23. And each of those chromosomes, each of those unique chromosomes has a name. So chromosomal names in humans, the autosomes are named by numbers, and they're numbered 1 through 22, and that's from the longest to the shortest. So the longest human autosome is chromosome 1, the shortest is 22. In different species, they would have a different number of autosomes, but the convention is still the same. And we also have in humans and other mammals, the sex chromosomes, which have the names X and Y. Now, we can also name or refer to regions within the chromosomes. And so chromosomes, each chromosome has specific regions. And those are defined by what are called G-bands. G-bands are the result of a special staining. So this is kind of like the old school staining of chromosomes by a dye called Giemsa. So if you Giemsa stain chromosomes, it results in a light, dark banding patterns, where the darker regions pick up more stain. They keep more stain, and these are heterochromeric. So these are more densely packed regions of the chromosomes, and then the lighter regions are euchromatin. They're less densely packed. So here's a cartoon depiction of sort of the banding patterns that we would see in the different chromosomes. So here's chromosome 1, and this is the banding pattern. So if you stain this with Giemsa, you would get a similar banding pattern, and all chromosomes 1s, all normal chromosome 1s would have this same banding pattern. So the banding pattern doesn't change. So that heterochromatin, euchromatin regions can be depicted here in this. And you can see that each of these has a region. And those regions are numbered starting from the centromere outward. Write this down. So the G bands form the banding patterns, and these banding regions are numbered starting from the centromere outward. So if we look at that, that means that we actually have two sort of region, or not regions, but what we call these are arms. So we actually have two arms of the chromosomes. And they each have their own region numbering. So the arms are created by the position of the centromere. Arms are created by the position of the centromere on a chromosome. And if you look at this little image again, what we see is that the position of the centromere is fixed. This is where the position of the centromere is on chromosome 4, chromosome 5, and that's fixed. But you notice that there's never a centromere that's right exactly smack dab in the middle. So these arms that are created, one's always smaller and one is always longer. We call the smaller arm the petite arm or the P arm. So the P arm is the petite, or the short arm is more commonly used. And then the Q is the long arm. So it's not P, it's the long arm of the chromosome. So each chromosome has a P arm and a Q arm, and they each have their own regions. And the numbering of those regions starts from the centromere going outward. And we can also classify chromosomes based on, again, that position of the centromere. So types of chromosomes based on the position of the centromere. Chromosomes that have centromeres that are just about in the middle, it's never exactly in the middle, are called metacentric. So metacentric have the centromere about in the center. But never exactly. Actually, I think I have an image here, so let's take a look. So here's a metacentric chromosome. We still have a short arm and a long arm. So metacentric has a centromere about in the middle. The submetacentric is somewhat off center, so it's a little more off center. Making that P and Q arm a little more distinct. We have an acrocentric chromosomes. So acrocentric chromosomes have a centromere near the end. So the centromere is near the end of the chromosome. So the P arm is almost negligible. So there's very little P arm. And then you have, and in humans, we don't have this, but a true telocentric chromosome has no P arm at all. So a telocentric, the centromere is at the end of the chromosome. And so there would be no P arm. As I said, in humans, we don't have that. If we look here, we can see 13. This is definitely an acrocentric. You know, it's very negligible genetic information there, but there is a small P arm. This one here would be submetacentric. I would call this one metacentric, this one metacentric. So that's how we refer to chromosomes based on the position of their centromeres. And we also have some genetically unstable anomalies that we're going to talk about. So some unstable anomalies. Chromosomes that have no centromere, and we call those, not to be confused with acrocentric, but acentric. So no centromere. So these are chromosomes with no centromere. We'll find out those are very genetically unstable. And we can have a situation where we have two centromeres on a chromosome, and those are dicentric chromosomes. They have two centromeres. And these are also very unstable genetically. So we can refer to chromosomes based on the position of the centromere. We've got regions of each of the P arms and the Q arms. So we can also name them. So if each gene has a physical locus on a chromosome, we can name the locus. So naming the gene locus. I guess this is maybe best done by example. So I'm going to give you the ABO blood type locus. So the gene that codes for that branching enzyme, that carbohydrate branching enzyme that determines your blood type, is located at 9q34. So this is chromosome 9. So the chromosome number comes first. The arm of the chromosome comes next. And then this is the region in the band within the region. So let's see if we can find the ABO blood type locus in this little cartoon karyotype here. So chromosome 9, it was 9q, so it's in the long arm. This is region 3, and this is 34, right? So 3, 4, this is where we would find the ABO blood type locus. Here's another showing the nomenclature. The chromosome number comes first. This is saying it's on the short arm of region 3, band 1, and then you can even get like subregions within the regions. So that's how we define a locus, but how do we define a karyotype? So that complete chromosomal composition also has nomenclature. So karyotype designations. Remember, karyotype indicates the total chromosomal composition in an individual. So it's the chromosomal composition. So in a human, a normal human, so if there are no chromosomal abnormalities and it is normal chromosome content, a normal human, the karyotype would be depicted as 46, XX, or XY. So a female would be 46XX and a male would be 46XY, where this is the total chromosomal number. And included within that total number is either XX or XY, but then it's the sex chromosome composition is indicated. Well, what happens, how do we indicate if we have abnormalities in a karyotype? Because that's where we're going, right? We're going to chromosomal abnormalities, abnormalities in chromosome number, abnormalities in chromosome structure. So there are some common symbols we can use. I'm going to show you a chart. Here's a chart, again, talking about the symbols and the nomenclature that have to do with the chromosomes. There are two common sort of symbols that I want to point out when we're indicating abnormalities. So if we are talking about, so abnormal karyotypes, so some common symbols and indications we would use in an abnormal karyotype. If we had a chromosomal addition or loss, so loss or gain of an entire chromosome, we would, these are actually called somes, and we will talk about the somes, but we put a minus or a plus sign respectively. If it's a loss, it's a minus. If we're gaining a chromosome, it's a plus before the chromosome that's added or gained, before the number of the chromosome that's added or gained, or lost or gained. So let's see, what does this look like? So let's say we're going to add a chromosome 21 to a karyotype. So someone picks up an additional chromosome 21. Well, we still put the total number of chromosomes first. So if you've got 3 chromosome 21s rather than 2, what is the total chromosomal composition? It would be 47. And let's say it's a male. So we'll say 47 XY, and then you indicate the abnormality. In this case, it would be plus 21. You might know what this is. This is also called trisomy 21, otherwise known as Down syndrome. And we'll touch on this again when we get into the abnormalities. So this is trisomy 21. You've got an extra chromosome 21, so it's indicated like that. If you lost a chromosome, then it would be a 45 sex chromosome composition minus whatever number of the chromosome that's lost. Now we can use these minus and plus symbols in a different way as well. So if we are not losing or gaining an entire chromosome, but let's say we're having a duplication or a deletion, or let's say a deletion or a duplication within a chromosome, we can use that minus and plus again. So I'll put minus and plus respectively, but we don't put it before a chromosome number. We put it after the chromosome that has a deletion, after the region that's deleted. So let's look at an example of that. Let's say, here's an example. Let's say 46, because we're not losing or gaining an entire chromosome, XX 16P minus. Okay, so what is this telling us? We've got normal chromosomal numbers. It's a female who has a deletion in the short arm of chromosome 16. So this is indicating a deletion in the short arm of chromosome 16. Now, if the entire short arm was deleted, we would say 46XX 16 minus P. That would be entire short arm deleted. And sometimes DEL is used to indicate deletion, but so this is just some common terminology. If you had a duplication, you would use a plus rather than a minus. And so the position of that plus or minus is important because this is a deletion. This is an addition of an entire chromosome. As we move towards these abnormalities and looking how they affect an organism, we're going to find out that a key thing that is going to affect and impact the organism is the gene dosage. If we unbalance, if we mess up the normal gene dosage in an organism, it's usually very, very destructive and not well handled. So what do I mean by gene dosage? Genes don't do anything, they're recipes, but they're recipes to make proteins, right? So a gene dosage is the amount or the concentration really of the gene product produced. And the gene product in this case is protein produced for each gene. And it's proportional, so it's normally the amount of protein that's made, it's proportional to the number of alleles. For each of the genes. So normal gene dosage in a eukaryotic organism or in a diploid organism is a diploid, is 2x essentially, right? So an organism, the number of sets of chromosomes, the number of alleles for each gene that an organism has is called its ploidy. So the ploidy is the number of complete sets of chromosomes. And we've seen some of these terms before. We know that haploid means one set of chromosomes, therefore one allele. In a haploid cell, the normal gene dosage then would be 1x because you only have one allele for each gene. In a diploid cell, we have two sets of chromosomes, therefore we have two alleles for each gene, therefore we would have 2x gene product, or 2 times the protein made in a diploid cell than we would in a haploid cell. In some organisms, not humans, not animals, animals don't handle this well, but in the plant kingdom, we can have polyploid organisms, which means greater than 2 sets of chromosomes. So some examples, triploid, you can probably guess, would be 3 sets. Tetraploid would be 4 sets. So this is more common in plants. And often the greater the number of chromosome sets, the bigger so greater the number of sets, the bigger the plant in the fruit. So here's just a depiction here. In a haploid organism, we've got 1 of each. Diploid, we've got 2 of each. Triploid, 3 of each. Tetraploid, 4 of each. Now, if you have complete sets, if each chromosome is in a balanced number, we call that euploid. Or euploidy is when you have a balanced sets of chromosomes. Therefore, you also have a balanced gene dosage. So if we take a diploid organism, each gene has 2 copies. So for each gene in the organism, you've got 2 copies, 2 alleles being expressed. You've got a 2x gene dosage. In a triploid, now you've got 3 copies of each allele, but all of the genes are in 3 copies. So they're balanced, right? They're all in a 3x sort of situation. Tetraploid, you've got 4 copies of each allele, but all of them are in 4 copies. So it's a euploid situation because it's balanced. What we're going to find out is this is good. Organisms like to have balanced gene dosage. And when it's not balanced in an aneuploid situation, aneuploidy is an abnormal number of chromosomes. So this is when you lose or gain a chromosome. These are your somes that we talked about before. So let's look at a diploid organism. What if we gain this extra one here? This would, every gene on this yellow chromosome would be in 1, 2, 3 copies. That's going to unbalance the dosage. And this is really super intolerable to organisms, whether it's animals, plants, bacteria. Gene dosage is super critical to sort of viability of an organism. If we think about humans and we think about mammals for a second, does anything when you're thinking about your chromosomes come to mind like, wait a second, 2 copies of all of them, 2 copies of all of them. Is there anything that's bugging you, like kind of nagging you at the back of your mind? Is there a situation where you don't have 2 alleles for each gene? Think about a human male. A human male is XY. That means all the genes on the X are they only have 1 allele for that. So in males, you only have 1 allele for each of the genes on X. In females, you have 2 alleles for each of the genes on X. But the proper dosage for the sex chromosome genes is actually 1 allele. And so for the sex chromosomes, let's see, where was my, for the sex chromosomes, a process called dosage compensation occurs. Dosage compensation, and this is an equalization of the gene products. And in humans and animals, all but one X chromosome undergoes a process which creates hypercondensation of the X chromosome, of one of the X chromosomes, and it can't express the genes. So all but one X chromosome is inactivated. The process is called X inactivation. So X inactivation occurs in human females and mammalian females that have two X chromosomes. So in females, in XX females, at a very early stage of development, so actually before it's phenotypically a female, but they have two X chromosomes, at a very early stage in development, and that stage is like pre-embryo. It's at the 64 to 120 cell stage. So you've got, here's your egg, here's your sperm, making your zygote. So this is going to divide from one cell. We've got 2, then we go to 4, then we go to 8, then we go to 16, right? Mitosis, mitosis, mitosis. When we get up to the 64 to 128, so let me just kind of, it's actually this blastocyst stage. And so you've got, let's pretend like we've got 64 cells in there. They kind of mass, they have what's called the inner cell mass. And at this stage of development, which is really like 3 days after fertilization or so, at this stage is when one of the X's is randomly selected to become inactivated. So at this stage, one X chromosome is randomly inactivated in each cell. And so let's say in this one, the male, the paternal X is inactivated. And let's say in these ones, the maternal X is inactivated. The inactivation process actually occurs by hypercondensation. This is hypercondensation of the X chromosome. And therefore you get decreased, or you lose gene expression from that hypercondensed X. The physical structure of this hypercondensed X is called a bar body. And you can see this if you stain female chromosomes at a certain stage of development. You can see this hypercondensed X chromosome because the other one is going to be not condensed. So here, so notice that in 50% of the cells-ish, the paternal X is inactivated. In the other 50% of the cells, the maternal X is inactivated. And that is the same one that is going to continue to be inactivated throughout development. And so let's take a look here at, here's what a bar body would look like in a nucleus of a stained cell. Here's the process of X inactivation.
The cells start out in a female with 2 X chromosomes. The cell divides and the embryo is getting slightly bigger, and still each cell has both X chromosomes active. But in early embryogenesis, each cell will inactivate one of its X's, and one cell will remain with the paternal X active, while the other one, the maternal one. And now this process will happen at random, and you'll have almost half of the cell with the maternal X active, half with the paternal X. The embryo will continue to grow, cells divide, all the descendants of that cell, all the descendants will keep the same X active. And you've seen this in calico cats, right? The cross-colored gene is on the X chromosome. That's precisely why these cats look like this in mosaic appearance, because of the process of X chromosome inactivation.
So the process of gene dosage of X inactivation balances the gene dosage of the sex chromosome. So the normal sort of gene expression, gene dosage we should have coming from the sex chromosomes is really 1X. We should have one allele actively expressing the gene. And that's the inactivation, this hypercondensation of one of the X chromosomes significantly reduces expression from that X chromosome.